I actually think of it as the same system. The immune system and the microbiome go hand in hand, right? In fact, the immune system would cease to function if it wasn't for your microbiome. So you might have all your glands, you might have your bone marrow, you might have the capability of producing all the immune cells that you need, right? Your T cells, your B cells, your dendritic cells, macrophages, and so on. You can produce all of them, but they learn how to function from the microbiome. So what they showed in the study, and this was a series of animal studies, is that they could take a mouse that had all of the immune components, including dendritic cells, but no microbiome, right? So they would raise these mice, they're called no-biotic mice, under sterile conditions. They would hit them with antibiotics early on, so they're relatively sterile, and then they have no exposure to microbes. So they have all their immune parts, but they have no microbiome. When they inject viruses into these mice, the immune system is there, but it doesn't respond at all.
I started sending away for stool tests before Genova was even great. It was Great Smokies, and even before that, back in the 80s, I sent my first stool tests off to I forgot the name of the guy who started Great Smokies back then, that is now Genova. And they have been a source of great help and great confusion ever since. As you know, we've gone a long way from just culturing the bugs to first using RNA and now next-gen sequencing to really get better and better look at what bugs are in your gut. But unfortunately, as the knowledge of the incredible variation in gut flora is out there, the knowledge of what to do about it is struggling to keep up. And I'm very confusing to practitioners and patients, because how do we protect this microbiome? And I think most important thing, it's a very resilient, just like we are incredibly resilient. And I know that, we worry when we disturb our microbiome, and we have great concerns, and we should have them, about the quality of food, what we feed the microbiome, what we do to it with antibiotics, and just the herbicides and pesticides that contaminate our food. I mean, these are all big concerns and real ones. But I just would also like to temper that with the thing is that will remind us again and again how resilient we are. And because we treat Lyme disease, we have seen people who've had really distressed microbiomes. I mean, like where, for instance, many of the labs do, and Dr. Krishnan's lab, the microbiome labs, they look at anywhere from 10 to 15 of what people call Keystone species. These are the species in the gut that we have found seem to support a healthy environment. They produce chemicals that allow the other bacteria that we need to thrive. And even though these but these are different in different groups of people, different populations, different diets, different genetic backgrounds. So it's not that the Keystone species are all universal. Some of them probably are, and that's something we're going to ask Dr. Krishnan about. But the important thing is, if you have had a bad diet for a while, if you've had to be on antibiotics, all is not lost. Okay, I think one of the things that I find disturbing when I listen to many talks, or even just when I'm reading, it would sound like, oh my God, my microbiome is going to be destroyed because they gave me antibiotics when I had ear infections as a kid, or because I took antibiotics now as an adult when I had a bronchitis or a sinusitis, or I was on them for months when I had Lyme. Oh, good morning. So that's my soliloquy. Good morning, Krishnan.
Hey, how are you, Eric? Good to see you.
Very good to see you, Kiran. I was just giving a brief, not an introduction, I was giving a brief, well, not quite a rant, but a brief little intro into my feeling of too many people, I think, get too worried that they're going to destroy their microbiome if they transgress a little bit, you know, and yes, it's much better to have a pristine system. We're all do well with that, but, you know, life is life and we're not going to be perfect, and don't give up hope because you had a course of antibiotics along the way.
Absolutely. So. Yeah. The microbiome is resilient. And, you know, given, I always look at it as like 80% of the time, it's good decisions and it's going to be fine. You know, I think we all have at least 20% of slack to play with, right? So, and microbes are resilient, right? They can come back if the negative impact is not super persistent over time.
Yeah. And that, well, so welcome. I should introduce you formally a little bit. Dr. Kiran Krishnan, who is a microbiologist and who has, luckily for us, taken his knowledge and helped create a company and several companies that help teach us, learn, and treat the microbiome. So, I mean, really has really opened my eyes to many aspects of how important the microbiome and what it can do. And one of the things I'd like to just start off with just briefly, and we don't have to spend too much time with this, but this is more, I think, for me than our audience, is the technologies behind the testing, you know, because that's what's confusing to a lot of people, is why one test gives me this results and another test gives me different results.
Yeah. And, you know, the microbiome testing is a bit of complex technology. It's a good mix of laboratory science techniques plus computing capabilities, right? So it requires high computer output as well. And I want to say hello to everyone. I see messages popping up from Finland and other places. So thank you so much, Dr. Gordon, for having me. And this is a pleasure. So so let's talk about microbiome testing, right? So in humans, we have about 22,000 genes in our chromosomes. So when we, you know, take those cheek swabs and things that we use to do our own genetic testing, we're dealing with about 22,000 genes that are functional. When you take a sample of the microbiome, you're getting upwards of 2.5 million different genes, right? So the numbers are much more profound when it comes to the microbiome. And bacteria will share lots of similar genes within one another. So then how do you properly distinguish the microbes from one another? Not to go too much in detail, but there's basically two forms of sequencing. There's something called 16S ribosomal RNA sequencing, and there's another thing called whole genome sequencing. I'll take the 16S piece for a second, right? So within any bacteria's genome, they tend to have 6 or 7 regions, where if you have all of those regions together, it denotes a particular bacteria, right? So imagine bacteria A has 6 or 7 unique regions that are combinations of genes that the microbe has. Now, in 16S sequencing, what researchers used to do is chop up the DNA, and then they'll use probes to look for those 6 or 7 regions. And if they find all 6 or 7 regions, then they can say with some degree of confidence that you have that microbe present, right? So you can identify that microbe is present in the environment you're testing. Now, here's a complication. When you have hundreds or thousands of microbes in a given sample, and many of those microbes may share regions, right? And those regions being so small in a giant milieu of genes, it may be hard to pick up all of those regions, right? And because it's hard to pick up all those regions, there are programs that have been developed to pick up 2, 3, maybe 4 of the 6 or 7 regions, and then make a guess as to what microbe that is based on an algorithm. So it's a guessing game. It's a statistical guessing game, which brings down the accuracy quite a bit. So the 16S technology was really useful early on in the world of microbiome education, and it's good for looking at groups of bacteria rather than down to the species, right? And so now we have something called whole genome, which looks at the entire genome of the microbe from end to end, thereby being incredibly accurate. in terms of showing and detecting which microbes are in place. So if anyone's getting a stool test done, there are two things to consider. Number one is the stool test using whole genome sequencing? If it is, then it's the most accurate way of doing it, right? If it's not using it, then it's a big guessing game. Number two, the sequencing part basically gives you tons of genetic information, right? It's terabytes of information from a single sample. Then that information has to be assembled in a usable way. So it requires what we call bioinformatics. And it requires really well-established computer programs to take all of that information and make any sense of it in which it impacts your health. So having experience in doing that and having the right databases to plot against is really important. So those are two important considerations when you think about doing gut sequencing.
Yeah, and I'm just reviewing some of the literature. It's the computer work is kind of mind-boggling.
Oh, it is.
But you're constantly having to find a needle in a haystack and put it in the right order. I mean, that's mixing metaphors, but it's just amazing that you have been able to do that and to get the information. And so now when we have all that, okay, so now we're in that place we have almost too much information, like we have to then decide what to do with it. And I have been fascinated by the Keystone species idea that maybe, we can help, that can guide us. But even amongst them, there seems to be lots of variability about which Keystone species are really Keystone for whom, if you will. So
there are some uniformities, right? So
yeah,
of course. So you think about some of the most well-studied Keystone species. So you have organisms like Faecalibacter prosnitzi, Akkermansia muciniphila, Bifidolongum. These have been shown in large number of studies to be generally beneficial to the host. And in other studies, they've delineated what exactly the mechanisms are of the benefit, right? So in general, if you have elevated or proper levels, detectable levels of these organisms. So I won't say elevated because the healthy range can be different for different people. But if you have them present and you have healthy levels of them, then they're going to be affording resilience in part, which is what we were just talking about. How much slack can you give yourself in terms of the choices you make and so on, is largely dependent on how resilient the microbiome is. And the resilience of the microbiome is based on how many of these species do you have and at what level, right? And so,
No, I was just going to say, just because we sometimes forget, I didn't know, the Keystone species, I think I mentioned earlier, are just species that, correct me if I'm wrong, that kind of support the environment. They're the ones that if they go away, the environment really might crash. And they also produce chemicals that the other bugs need to grow and to survive. And if you could even make that better, explain that even more fuller, it would be great, just so people really get. Yeah.
Absolutely. So, and that's why they're called Keystone for that reason, right? So, and the term Keystone comes from, if you look at architecture with the arch, that stone in the very middle is called the keystone. And the reason is because once you put that stone in, then all the pressures are aligned and the arch is absolutely stable. So it's the same analogy. These organisms provide stability to the microbiome, and they actually are inversely correlated with disease process in the host. And for a couple of reasons. So we'll break those down. Number one, in terms of how they provide stability for the microbiome, they control, for example, the pH of the gut, right? So they produce lactic acid as one of the functions that they do. They also help convert lactic acid into things like short chain fatty acids like butyrate, propionate, acetate, which feed the lining of the gut, which feed the cells that line your gut. It supports the immune system. It feeds the immune system with energy. It actually brings down inflammation and affects a lot of metabolic processes that are occurring in the gut. Many of them can produce secondary byproducts that actually feed other microbes like food. And so by doing all of those things, by managing the immune system, managing the lining of the gut, providing a nice thick mucosal lining, which is where all of the bacteria in your digestive tract live, and it's in that mucosal lining. So by providing a healthy mucosal lining and by affecting certain metabolic processes, they do a really good job of maintaining the overall health and stability of the microbiome. Now, they also have direct impact on the host, because some of these organisms produce compounds that the host will directly use, right? So Akkermansia, for example, can produce urolithins. Urolithins are a compound that's metabolized from polyphenols that are coming in from the diet, and then the bacteria convert the polyphenol into a compound that is really important for your mitochondria. So it really helps regenerate your mitochondria, turn over broken and damaged mitochondria, allow your cells to function more optimally. That way you can function more optimally overall, right? Remember, mitochondria are the little engines in the cell. So these keystone species, this is part of how they are associated with overall health, is because not only do they maintain the microbiome and the population there and help keep diversification and help maintain resilience, they also maintain certain physiological structures of the host, the lining of the gut, for example, the mucosal lining, the tight junctions, and so on. And they also produce compounds that the host will directly use for benefit.
Yeah, I think that's what's the amazing part of the gut, is that, how we keep learning how important, how many new functions that they are serving us, or again, I guess it's just the reality that we are not, that we are not us. we are an amazing conglomerate of bacteria and viruses and fungi, and it's just it's mind-boggling to realize the complexity, and I think that is overwhelming. But luckily, we have we can hook ourselves onto small areas and think and get useful information, and that's where because we see in our patients how many people are have lost a bunch of their keystone species because of diet and exposures. And how important it is to try to restore that. When you can you talk a little bit about I'm really interested in the effects on immunity and the effects on stress, because those are two big areas that our patients are dealing with all the time, is trying to get their immune system to function a little bit better, and how to deal with stress and sleep. And what does the gut have to offer for that?
Yeah, absolutely. So, and in fact, by asking that question, you actually create a good segue, because both immunity and stress and mental function as it relates to the microbiome are actually all connected, because stress response and sleep capabilities related to how the immune system is responding to signals in the body, and the microbiome plays a very important role in what signals those are and how those responses are dictated, right? So I'll break down that a little bit for the audience. So when you think about the immune system as it relates to the microbiome, I actually think of it as the same system. The immune system and the microbiome go hand in hand, right? In fact, the immune system would cease to function if it wasn't for your microbiome. So you might have all your glands, you might have your bone marrow, you might have the capability of producing all the immune cells that you need, right? Your T cells, your B cells, your dendritic cells, macrophages, and so on. You can produce all of them, but they learn how to function from the microbiome. There was a series of studies that were really interesting and actually published in March of 2020 that showed that your dendritic cells, for example, these are really important monitoring cells, right? So these cells are the early responders to the presence of an infection or toxic agent. They'll go out to the site of action and actually swallow things and grab onto full bacteria, full viruses. They'll engulf them, and then they'll present components of these bacteria and viruses and all to the rest of the immune system, so you can activate your T cells and B cells and so on to go against whatever that target is. So this is an antigen presenting cell. which is a really, really important cell. If those cells aren't functioning in your body, your immune system is really, really compromised, right? So what they showed in this study, and this was a series of animal studies, is that they could take a mouse that had all of the immune components, including dendritic cells, but no microbiome, right? So they would raise these mice, they're called no biotic mice, under sterile conditions. They would hit them with antibiotics early on, so they're relatively sterile, and then they have no exposure to microbes. So they have all their immune parts, but they have no microbiome. When they inject viruses into these mice, the immune system is there, but it doesn't respond at all, right? The virus is there infecting the mouse's cells, and the dendritic cells will just sit by and do nothing, right? Because it doesn't have the activation signals. And then when they add in the microbiome into these same mice, all of a sudden the dendritic cells start responding. And then you start going, okay, how is that? What is the mechanism there, right? So at a simplistic level, basically what it is the microbes in your microbiome have dictated what the activation threshold is for your immune system, right? And this is really important to note, because keep in mind that the immune system, for the large part, are monitoring and looking for microbes, right? In a system that is covered with microbes. And so the job of the immune system is really difficult. The analogy I give to put numbers in perspective for people, is that it's about a 200,000 to 1 ratio in any given part of the body where you have monitoring immune cells that are looking for infectious agents and so on. You've got 200,000 benign microbes in that space for the one immune cell that's monitoring the space, right? So when you think about that, the analogy I give is that imagine you're at a music festival that's contained in a given stadium. And in that stadium, there are 200,000 individuals, right? That's a lot of people. And among the 200,000 people, there's one individual that has, ill intentions in that space, right? They plan to do something bad. And you are the one and only security guard in that stadium. And everybody looks about the same. So how would you in any way find that one individual or survey for that one individual, right? The only way you could do it is if the other 199,999 people were on your side and were playing neighborhood watch, and they were all radioed into you looking for someone that's doing something suspicious. And the moment they see something, they'll radio you and tell you exactly where it is. And that's what the microbiome does for our immune system, because every part of your body on the inside is covered with microbes, right? So the first organisms to notice any change in the environment from a new virus or new bacteria, a new fungus or something that comes in are the surrounding microbes. And then they send signals called interferons or interleukins to activate the immune system to come and attack in that region. Now, the reason this exists is because what is as bad and maybe arguably worse than an immune system that is not responsive is an immune system that's overly responsive, right? Which means that the immune system is constantly attacking all the microbes in your system. That means you're going to be sick all the time and you probably won't survive very long. So there's this relationship that has been developed where the microbes go, hey, we'll be your eyes and ears, we'll be the neighborhood watch. The moment we see something wrong that shouldn't be here, then we'll signal to you, you show up, you attack what's the thing that's the problem, and then they build this symbiotic relationship. Right? So that's the microbiome and the immune system. So we really cannot talk about the immune system without addressing the microbiome. And it's very clear that if your microbiome is compromised, your immune system doesn't function the same way. There's two great areas of study of this. One is in cancer treatment. So if you look at immunotherapy for cancer, immunotherapy can be a very powerful tool in cancer because it basically uses your own immune system to go after the tumors, right? And if it works really well because your own T cells can find the tumors, get rid of them, and then it almost never comes back because now your T cells are trained to look for that tumor, and it doesn't damage anything else in the region, right? So it's not the same as radiation or even chemotherapy that can damage lots of stuff. The thing is, studies have shown that if you had a course of antibiotics before starting immunotherapy, the effect of the immunotherapy is significantly lowered, right? Or upon microbiome analysis, if you have low levels of Keystone species, and you have low diversity, the probability that the immunotherapy will work well is lower dramatically. right? And this was also seen in COVID, because we all came to understand our immune systems when COVID hit, right? There's a number of studies on COVID as an acute disease, and then also people with long hauler syndrome, where they've mapped what the microbiomes looked like in people that had very severe response to COVID and even death, and then also people who developed long COVID. And there's a very clear microbiome signature. The people who had the worst outcomes for COVID had high pathobiomes. That means that you had high levels of potentially pathogenic organisms or opportunistic organisms, and low levels of certain keystone species like Faecalibacter prausnitzii, like Bifidolongum, and most of those people tended to have low diversity as well, right? So it's pretty clear that if your microbiome is compromised, it's very hard for your immune system to function the way it should to protect the host.
Yeah, no, that's, there was, we've been looking at this for a long time. There were books, I mean, before we had such good explanation of the microbiome, a lot of the focus was on parasites and how important that was. There's a wonderful book called, The Epidemic of Absence, about how, like the fact that as we in the Western world, kids are not exposed to, parasites, to dirt when they're young, especially in that first few years of life. And we just don't get the exposure to all these bacteria, parasites, viruses that should be at least passing through the gut, if not hanging out there, and educating our immune systems. Because as you said, when the immune system stays dysregulated, that's when people get chronic illness. That's the people that, a lot of the people on this webinar are patients or people who followed some of our webinars, and it's all about chronic disease. And to me, chronic disease is essentially chronic inflammation. The body's not able to turn off the self-defense mechanism. And this is why the gut is always been central. But every few years we come back and realize, oh my God, it's even more important than we realized.
We learn more and more details, and we're like, holy cow. And what you just said, the inflammation and the gut, both of those are the key things. And then that actually is a good segue into the stress component that you asked about, right? So one of the things that is absolutely true and inevitable is we're all going to experience stress, right? Life is stressful in general. Even if you have a great life, even if everything's going the right way, for the most part, humans are going to find things that make them stress, right? So, and when we're stressed out, we trigger something called the HPA axis, which is the hypothalamus pituitary adrenal axis. This axis has a number of control points, but the end point of the axis is twofold. Number one, it's the activation of the adrenals to release cortisol, which is the well-known stress hormone that most people are familiar with. And then at the same time, it's also activating the sympathetic nervous system, which is the part of the nervous system that gets you ready for flight or fight, right? So make no mistake, when you experience stress, and you experience the warming and the increased heart rate, and increased respiratory rate, and, that feeling in the back of your neck and so on, that is all your flight or fight response, right? And the flight or fight response is extremely important. It's ingrained within us because that was a key component to surviving through the course of evolution, right? We humans evolved in a harsh world of predators and competitors and things like that. And so they had to be very acutely aware of their environment. And should there be a signal of danger, physiologically, we have to be ready to fight or flee from that danger, right? So what happens when you experience a stressor, and even though the stressor nowadays often aren't life-threatening like they used to be, it could be just an email or a tweet, or someone cuts you off in traffic on the highway, it's still activating that same biological mechanism as if there was a predator like this behind you following you around, right? And so so the same biological mechanism is going on. So let's talk about what happens there. So your HPA axis, your hypothalamic pituitary adrenal axis, gets activated. Your hypothalamus picks up the external stressor first. A series of hormones are released to get your adrenals to release cortisol. Now you start getting cortisol building up in the body as you're developing more and more stress response. The sympathetic nervous system at the same time is trying to increase blood perfusion to your heart, to your brain, and to your muscles, right? Because it wants your brain to be very acutely aware, so you can increase visual acuity, auditory acuity, so your sounds and sights are much more clear. You need blood to your heart so that your heart can function properly to start pumping blood to your muscles so that you can fight or flee. Now, the way your body does this is by using the immune system. It gets blood to your brain, so your central nervous system and your heart, your muscles, through the immune system by activating cells like those dendritic cells I talked about earlier, right? So macrophages, dendritic cells, microglia cells are the cousins of those cells in your central nervous system. So those cells, when they start getting these stress hormone signals and they start getting signals from the sympathetic nervous system, they start releasing all these pro-inflammatory cytokines in your brain, in your heart, in your muscles, so that you get more blood perfusing to those areas. So make no mistake, when you're stressed and when you're in that starting stages of flight or fight, you are inflamed heavily in your brain, in your heart, and in your muscles, right? That is one of the outcomes of that. And then as cortisol starts to build up, some of it gets dumped into the gut. And one of the reasons why it gets dumped into the gut is because part of that cortisol gets metabolized by organisms that then send the metabolic byproducts to the kidneys, where they activate sodium-potassium pumps to bring in more sodium and potassium into circulation, which increases blood pressure. That pressure is also needed to get more perfusion. This is why chronic stress can create hypertension, right? Because you have these pumps open all the time, and you're creating this hypertensive situation. And so what's happening there is that you're supposed to go through this cycle, right? And then you're supposed to be able to fight or flee from the danger. And when you go away from the danger, you're supposed to be able to come down from all of this, right? The inflammation goes down, the sympathetic activation goes down. You start turning on the parasympathetic side of your nervous system, which is the rest and digest and recover part of the nervous system, not the flight or fight system, right? So you're supposed to go through the cycle and come back down. And the way you come back down from all this is cortisol still. So cortisol keeps increasing as you're going through the stress response, but as it keeps increasing, it'll start binding something called glucocorticoid receptors. These are receptors that bind cortisol on many different cells in the body. Virtually every cell in your body, with the exception of maybe red blood cells, have these glucocorticoid receptors, including the cells in your central nervous system, in your hypothalamus, in your pituitary, in your adrenals. When they start binding cortisol at a certain concentration, it starts sending an off signal to turn this whole system down, right? So this is how you go up, you deal with whatever you have to deal with, and then you come down. And being able to go up and come down is perfectly fine. Here's where the gut comes into play, and this whole thing gets dismantled, right? As cortisol, as I mentioned, part of it ends up in the gut because it has to be metabolized in order to go to the kidneys to increase blood perfusion. If you have an unhealthy gut microbiome and you're missing certain species or certain features of your gut, when cortisol hits your gut, it creates a tremendous amount of permeability acutely. So your gut becomes very leaky very fast, right? We're talking about in seconds. And then because of that, you have all these inflammatory cytokines that go way up because of that leakiness. One in particular that goes way up is called interleukin-6 or IL-6. IL-6 is a big problem when it shoots way up like that, because one of the things it's been known to do and has been shown to do is reactivate the HPA axis as if there's another stressor. Right? So imagine cortisol is going up, some of it's dumping into the gut for that metabolic process, but what gets dumped in the gut causes leakiness in the gut. That causes IL-6 to go up, and then IL-6 goes to your brain, reactivates HPA axis, and releases even more cortisol, right? So you keep reactivating the HPA axis over and over and over again from a single stressor. That keeps you in a basal stress state, which means you are constantly in a sympathetic state, which is fight or flight, which means you have lots of inflammation in your heart, in your brain, in your muscles. And it also means you're not resting, digesting, recovering. So your gut becomes dysfunctional over time even more because you can't digest and break down food. So that's inflammation and the role of the gut microbiome. Now, if you have a healthy gut microbiome, you have species in there that can produce things like peptidoglycans and exopolysaccharides. These are specific carbohydrates that are produced by some of these keystone species like Bifidobacterium longum, they actually curb that inflammatory process that cortisol creates in the gut, thereby stopping that reactivation. And in fact, they can go as far as reducing inflammation in the brain, and they can go as far as shifting your brain waves. So you go from being in this beta or alpha, the high frequency, beta or gamma, the high frequency brain waves, which makes stress even worse. It makes you kind of frantic, and it starts to shift you towards theta brain waves, which is the more relaxing waves, right? And we have studies to prove this, that if you have these types of microbes in your gut, they shift your brainwave when you experience stress. So you can experience stress in a theta wave band, which is a very powerful meditative state, which doesn't activate everything. It's a low frequency state. It allows you to rationalize through the stressor and then overcome it pretty quickly.
Yeah, so balance. It's one of the things to remind people is there are, luckily, again, the redundancy of the body. There are many systems that will take you down and will help you relax and help this, because we are constantly stressed. We go in and out, but unfortunately, when the gut is, when we lose that gut, when we lose those species that we need there, we lose one of the brakes. Luckily, we've got a lot of brakes in the system. Otherwise, we'd be short-lived creatures. But we need to optimize all of them, especially when we get stuck in this area of chronic inflammation, which is also chronic stress, because as Dr. Krishnan pointed out, IL-6 is a big part of inflammation, and it also is ringing those alarm bells. So what are some of the ways that you help people, change their microbiome a bit? Because we want to get to some questions, but I want to get to some of that. You've done some very interesting work on help. We've laid out the problem. Now let's fix it.
let's get to the solution, right? Yeah. So probiotics can be extremely powerful for modulating the microbiome, but it has to be the right organisms, well tested, so you know exactly what they're doing in the system and how not only do they impact certain physiological responses, but then how do they impact the rest of the microbiome? That's a really important aspect of it, right? So we've gone through a lot of painstaking science, a lot of preclinical and clinical research work to isolate and figure out a few organisms that really help with some of these processes. And so probiotics can play a role. One set of organisms that we've worked on now for about 15 years are called spore-based bacteria. So these are Bacillus endospores. What's interesting about these organisms is that they're pretty ubiquitous in the world. You find them all over in nature, in soil, in rivers and streams and lakes and desert dust, in the polar ice caps. You find them, and they've been ubiquitous in Earth's environments for millions of years, right? So we have this beautiful symbiotic relationship with some of the species where we swallow them and we consume them, inadvertently, because for the most part during human evolution, people were eating off the land and were engaged in the land. So you gain significant exposure to these microbes. And so we give them a home. Our immune system doesn't see them as foe, doesn't amplify any inflammatory responses. In fact, welcomes them into the system and activates these spores. And when the spores get in, they start doing some cleanup job for us in our microbiome. One thing they're really good at is doing something called quorum sensing. Quorum sensing is where they read the microbial environment. They look at this bacterial, the chemical signatures of all these different bacteria, and they can identify dysfunctional or problematic bacteria. They'll sit next to those microbes and produce compounds like antimicrobials to bring down the growth of those dysfunctional bacteria, right? So they can, in effect, adjust the microbial environment in our gut, increasing diversity, bringing down dysfunctional microbes, increasing the growth of beneficial microbes, including keystone species. They also produce a number of compounds like short-chain fatty acids, like lactic acid, like peptides, all these different things, enzymes even, that help the function of the gut. And we also have shown in a couple published studies that they actually seal up the lining of the gut. We know that leaky gut is probably the biggest driver of chronic low-grade inflammation, right? So sealing up the gut and having really good tight junctions, good mucosal layer is a key part to having low levels of inflammation in the body. So this is just one subset of microbes that can do all of this stuff for you, right? And it's fascinating. We've published studies on acne on the probiotics. We've published studies on triglycerides on the probiotics. We have one coming out on rheumatoid arthritis, showing its ability to modulate a lot of processes in all of these conditions. All of them are inflammatory conditions, right? And so when you're able to bring down inflammation by sealing up the lining of the gut and modulating immune response, it goes a long way to helping with fixing the gut microbiome. The other one is I mentioned, the right type of organism in the gut to negate the leaky gut impact of cortisol, which then reactivates the HPA axis. What we found is that organisms that contain something called EPS or exopolysaccharide seem to conduct this function better than anything else, right? So if you have a really healthy gut, you'll have some bifidobacterium species that have exopolysaccharides on them. So this is a species that makes a very elegant and beautiful carbohydrate coating that they put around themselves. And it is actually this carbohydrate coating that does all the work of modulating the inflammatory response, of changing brainwave activity, of preventing the leakiness in the gut from cortisol, and from bringing down all of the stress hormones, right? So having that kind of species in your system is really, really powerful. We were able to isolate that from healthy volunteers. These were elderly Irish women who had a strong history of low stress, very good sleep, and all the stress and anxiety questionnaires, they scored very low. We did family histories and did family interviews and all that to understand that, yes, truly, they are very well balanced individuals and never really experienced stress the way other people do. And then when we went in their guts, we found that they all had high levels of these EPS strains. That was the only distinguishing factor between people who are the same age who had poor stress outcomes and poor sleep outcomes. Now, sleep is immediately tied because if you are stressed, you cannot sleep, right? If you're stressed, it's the sympathetic nervous system, the fight or flight system. Your body does not want you to sleep when you're in fight or flight because you're supposed to fight or flee, right? You die, and essentially, so until you get that parasympathetic turned on, it becomes really hard to sleep. So stress and sleep go hand in hand. So these types of microbes, which we have in a product called ZenBiome, they bring down cortisol, they bring down stress, they bring down the inflammation associated with it, they change brainwave activity, they lower the perception of stress, and then they start firing parts of the brain that are associated with vitality. They do all of this, and as a result, you also sleep better. So we have 2 published sleep studies on them as well. So those are some probiotic solutions that you can do, but there's other things you should add in as well.
Yeah, well, I mean, I keep coming, the redundancy of the system. There's so many ways, from breath to movement to light to, and so many supplements that will work for some people, but it's finding what your body needs. And, but always worth starting with the gut if you can. Many times you have to finish with the gut, but it's best to start if you're able to. But I think we've had, Darcy's telling me we have a lot of questions. So maybe let's see if we can answer some of.
All right, let me, okay, so the first one here, a few people have asked about how many primary keystone species we should have. And what's the best test to be requesting from our doctor to test?
Yeah, so, there are somewhere around 13, 14 known keystone species. And in the test that we do, it's called BiomeFX, which is a whole genome sequencing test. We test for all of them. And we look for something called relative abundance, which tells you the percent of that organism in your total microbial load, right? Which is the most accurate way of looking and understanding the context on everything that's going on in your microbiome. Now, do you need to have all those keystone species? Absolutely not. Almost nobody has all of them, right? Most people will have somewhere around 3 to 4 of the keystone species. We've seen lots of tests come back with just 1 keystone species. And they aren't necessarily suffering from much of anything, right? So there's a lot of functional redundancy within the microbiome. And so, if you're missing a keystone species, there may be a non-keystone species that can do a similar function, right? So actually the best measure, aside from looking at a couple of keystone species like Faecalibacter pause-nitzii, Akkermansia, Bifidolongum. If you have one of these three or all three of them, you're doing pretty good already. But the best measure that goes actually a step beyond keystone species is diversity, because diversity in the microbiome provides you with all of that functional redundancy, right? If you have 180 or 200 functional species in your gut microbiome, you have far more functional redundancy than somebody that has 110 or 115, right? So that's a thing to keep your eye on is what does my diversity look like? And then, what is the concentration of the keystone species I have? And you should hopefully have at least maybe 2 or 3 of those keystone species. And we use the test I mentioned it's called Biome FX, BIOME, the letter F and the letter X.
Yeah, I just as a note, so I am learning today too, because I always was hoping to see more keystone species, but so the diversity is definitely trumps most anything else. That's the most important marker.
Yeah, because of the beauty of functional redundancy in microbes, right? It often in a stable microbiota, if a microbe is missing or its functionality is missing, other microbes can develop that functionality if the need is there, right? One good example of that is there are people that are completely missing, the microbe that breaks down minerals and things that come into the gut, right? So these are I'm having a brain fart right now, missing it. I can't remember it. It'll probably come to me as I talk. And so if you're missing that microbe, then there's an increased chance of having things like gallstones and kidney stones and so on, right? Because you can't break down certain types of mineral complexes that come in. But it doesn't necessarily mean if you're missing that microbe, it's gone. That functionality is gone, because as it turns out, there are probably 2 or 3 other species that can do that, and they start to come back up because they don't have competition from that key microbe, right? What am I thinking?
Is it the one that does the glyoxylate pathway, or no?
It can, yes.
Yeah. I can't remember the name either, so
it'll come to me as we talk.
But I think that's just a beautiful thing, because what I like, when people have been ill for a while, like you said, we get stressed and we get more fearful. And I just didn't want the microbiome to be another source of great fear that, oh my God, I don't have enough. It's another reason I'm not going to get better. But there's so much to do. And again, nutrition is the basis, because if you don't start off there, unfortunately, I think you can take all the probiotics you want, and it's the milieu, it's the environment that supports the bugs to grow as well as each other. They need the right stuff. So let us some more questions before I keep waxing on about.
That's okay. All right, another question here for those AIDS patients who do better on low carbohydrate, few plants and vegetables, high healthy fats, and carnivore in general. Can you say anything about how their microbiome community of organisms looks? Certain species that carnivores have more of or tend to lack?
Yeah, so sorry, to clarify the question, that's if you're on a carnivore diet versus?
Yes, on a carnivore diet. Mm-hmm.
Okay, so there may be some short-term benefits being on a carnivore diet. And to me, a lot of that probably comes from cutting out sugar from your diet. But certainly increased protein intake. There are benefits to that as well. But any of those narrow diets over time will have a negative impact on your microbiome. And there are studies to show this now, that if you're on high fat diet for a period of time, I can't recall if they've done one specifically on carnivore diet, but a high meat diet, they've done it where over time, and we're talking about a year, 2 years, you'll start to reduce the diversity of your microbiome quite a bit, because a lot of the microbes in your large bowel that are, especially the keystone species, really need plant-based carbohydrates, fibers, insoluble, soluble, prebiotics, resistant starches. This is what they evolved on, right? They can metabolize some protein to a certain degree, but over time, you're going to get a reduction in diversity. So some of those, if there's a good clinical reason to use a diet like that for a short period of time, I would say, if that's the case, you'd want to really push getting back to a more diverse diet and increasing some of the intake of plant-based materials as well as you're moving forward.
Yeah, and I think this is a problem with diets, is that we learn about diets and we take them to extremes that no free living peoples ever did.
Totally.
Okay,
Yeah.
and that's the thing, is that nobody anywhere in this planet was just a carnivore. There was always lots of other stuff mixed in. You ate what you could eat.
Yep, absolutely. And even when they ate animals, they often ate like the digestive tract of the animals.
Yeah.
Contain lots of prebiotics and resistant starches and all that these animals, who are typically ruminants, have been, consuming and then ruminating. And so, yeah, like Eric said, there isn't a single obligate carnivore human, tribe or species out there. We are designed to eat a diversity in diet.
Yeah. I mean, even the tribes that are, I mean, they're mostly vegetarian. They eat lots of bugs, and whatever they eat. Everybody eats whatever they can when they can. Yeah. It's only in America we have people who become devout somethings.
Exactly.
And there's good reasons, but just be gentle with yourselves. This isn't the way the body was designed. We're supposed to eat dirt, folks. I mean, that's one of the things that I keep coming back to, is that this absolutely cleanliness thing in America. And I think you mentioned a study, the Finnish and Russian study. I don't want to waste too much time, but I thought it was beautiful that kids who are allowed to play in sandboxes had less allergies than the ones who were kept in their pristine environment. So it's the immune system needs information. Anyway, more questions.
Yeah, there's a number of questions about people that have had multiple courses of antibiotics, and what species and strains might be reduced. And also the other question that's coming up quite a bit is any species that might be reduced due to long COVID.
Yeah, so when you look at the microbiome data on long COVID, it's pretty clear that the individuals that develop long COVID have a type of gut to begin with, right? And that's actually what created the risk factor for developing long COVID. So it's not necessarily that long COVID has caused a reduction in any sort of species. It's the absence of certain species and the overrepresentation of others in the first place that gave you the risk of developing long COVID. As I mentioned, the people with long COVID tend to have an elevated pathobiome. A pathobiome is the component, the microbial component of your microbiota or your microbiome that are made up of organisms that have the potential of being pathogens. Right, so that's something we test for example, in the biome effects test. We give you a pathobiome score. Now, it doesn't necessarily mean that they're all bad and that they're causing infection and all that. It's very normal to have pathogens in the gut microbiome. The key is, what is the level of pathogens to the rest of the microbiota, right? Because the rest of the microbiota tend to keep the pathogens under control. And in some cases, there are even benefits from some of these pathogens, like training the immune system and so on. And so long COVID is represented by high pathobiomes and low levels of keystone species, especially Faecalibacter prausnitzii and especially Bifidobacterium longum, right? Yeah. Those two keystone are low in long COVID, and then the pathobiome is high in long COVID patients. Now, with antibiotics, it's difficult to say what organisms are suppressed after multiple courses of antibiotics, because it may be a little bit different for each person, and it's kind of different as to which antibiotic you took, right? So what are they gram-negative focus? Are they gram-positive focus? How long do they stay in the gut, and so on? But you can know this with a certain degree of certainty, is that after multiple levels or courses of antibiotics, you're going to have low diversity in the microbiome. Because inevitably what will happen is some organisms will come back faster than others with each dose of the antibiotic, which means that over a whole course, then you've actually favored the environment for certain organisms versus others. Diversity, the gut microbiome, is a measure of how many species, but also what is the uniformity of those species. Right, so low diversity not only means that you have reduced number of species, it also means that your species uniformity is low, meaning you've got a handful of organisms that make up 80% of all the population, and then another 150 make up the last 20%. Right, so that is also a skew in diversity, and that's one of the things that antibiotics do, is that each time you take an antibiotic, all the microbes go down in the first couple hours, right? And then they start to come back, but because of the changing environment, some will come back faster than others. And with each drop, you start to get a bigger and bigger differentiation between the microbes, right? Now, that being said, if you're thinking in your head, oh my God, 5 years ago, I had 4 rounds of antibiotics because of Lyme or whatever it is that you had, is it too late to fix your microbiome? Are those species gone forever from your system? The studies would indicate that that may not be true. They may not be completely gone. It's really hard to kill bacteria, especially in a complex environment like the gut, to the point where there's not a single cell left, right? This is even why, like cleaners, they even say kills 99.9% of germs. They never say 100%, because you can almost never indicate that 100% of germs are gone. And that's absolutely true in the gut as well. It's just that those organisms may be at such low levels that they're not really functional in the system. They're not providing, function to the host or to the other microbes. And that's where you're getting the dysfunction from. So adjusting the environment to allow them to start to come back up can be very, very potent. And you can do that through diet, diversifying your diet, lifestyle, so stress management, reduction of chemicals used in the home, cleaning chemicals, personal chemicals, personal care chemicals, and all that. Getting outside, getting in dirt, like Erica said a number of times, getting yourself on hikes and all that in natural environments, right? All those kind of things can help bring back these organisms.
Yes. My emphasis on dirt, it's just that, I just live amongst the world of this hyper cleanliness that has just, I think, caused so much problems in the young people over the last 70 years as it has grown. Mm-hmm. So anyway, it's just a big issue. So, but quick question, what about hand cleaners and things? Has that been shown to make a difference in the microbiome?
Yeah. So especially, like hand sanitizers. And in fact, there was a banning of a particular type of chemical that's used in soaps, right? These antimicrobial chemicals.
Triclosan?
Yeah, triclosamine. Yeah. And the reason for that is because they can be persistent on your hand. They can be, of course, they get into the water supply, and then now you have these antimicrobials in the water supply. They can be on your environment. These will have a huge impact on your overall outcomes, in part because we touch our face and all that quite frequently, right? And I always look at evolutionary behaviors and try to understand what the importance of those things would be. For example, human babies are very unique in a way because they sample their environment with their mouth, right? They basically, anything they can get their little hands on, they'll put in their mouth, right? Which is a very odd human behavior, because very quickly after we get to a certain age, we stop using our mouth as a primary examining tool, right? Humans in general are visual, auditory organisms, right? It'd be weird if somebody handed you a new object as an adult and you look at it and you go, what is this? And you start putting it in your mouth to sample it and figure it out, right? But babies do that all the time. So imagine our ancestral babies, when they're born, they're laid on the ground, they're putting dirt and dried feces and all that in their mouth, right? It becomes evolutionarily important to sample the environment around you with your mouth, and your hands are the big avenues of that, right? So your hands pick up microbes all over the place. And yes, unfortunately, because we live in relatively sterile worlds, you're picking up microbes from door handles and things like that, rather than from trees and bushes and natural environments. But nonetheless, these become a huge vector to get things into your system and gain exposure. So if you're constantly cleaning your hands and you're constantly sterilizing it, you're reducing that exposure. And then not only that, you're screwing up the microbiome on your skin, which then makes you more susceptible to things like eczema, dry skin, and all these other problems.
Yeah. No, it is, there is a balance, people, you will pass common colds and COVID, but on the other hand, maybe your body will respond healthier. We have to decide, how safe do you make it? When you take away all danger, you take away all life. Yep. It's a great dilemma. We have time for maybe one last question, I guess, if you have a quick one that you've noticed.
Yeah, just a quick one here. Can you hurt your system and skew the dominance by taking too many probiotics of one specific strain?
Oh,
Yeah. Okay. So there's this concept I've seen online about this developing a monoculture, right, in the gut by taking one probiotic for forever. That doesn't really happen. Think about your microbiome. It has something like 25, 30, 40 trillion organisms, right? That's a massive amount. Even if you're taking a probiotic with 50 billion, which sounds really like a lot, compare that to 40 trillion organisms that you're putting it into, right? So, and keep in mind, most probiotics you take aren't well adapted to exist in the gut because they're strains that were derived from other places and are grown under other conditions. So you're putting it in against 40 trillion organisms that are experts at colonizing the gut. So the ability of a probiotic to knock away a bunch of other organisms and create a dominance really doesn't exist. This has never been seen at all. But what is important to note, and we're seeing this more and more, in a conference we just did this past weekend, we presented some data on it where many probiotic strains are actually inflammatory. Right. And these are well-used common probiotic strains, Bifidobacterium, Lactobacillus, and so on. They all amplify inflammatory responses in the body. They do amplify some anti-inflammatory responses, but they amplify 8 or 9 inflammatory responses as well, right? And the reason for that is because the body doesn't see them as part of the commensal species. So normally when you send a bacteria in, your immune system is going to react to it, right? So automatically you'll get all kinds of inflammatory cytokines going up. Now, if you're a perfectly healthy person and what you're trying to do is train your immune system and just trying to upregulate it against the latest viral infection or something, that could be fine. But a lot of people listening and a lot of people that are being managed through functional medicine doctors and so on already have inflammatory conditions. And so the last thing they want to do is increase inflammation through a probiotic. So you do have to ask the question, is your probiotic silent? To me, that's a much more important thing because we're seeing data that many bacterial strains, many probiotic bacterial strains are not silent. When I say silent, I mean, do they not increase inflammatory response? And in fact, do they only increase anti-inflammatory response? So the strains I talked to you about today, the probiotic spores that we work with, we've shown that the Bifidobacterium with the exopolysaccharide, the anti-stress one, we've shown, or the anti-cortisol one, that's the one we've shown those to not have any inflammatory response at all in the body. In fact, it has very substantial anti-inflammatory response.
Okay, well, thank you, Kiran. It's just amazing. I mean, there's so much information out there. And I think, does your website have a lot of this for people to read and get more information?
Yeah, we do put out a good amount on microbiomelabs.com. That's labs with an S. Or if you reach out and follow me on Instagram, for example, my handle is Kiran Biome, so KIRANBIOME. I put out a lot of our research that we do, talks and information that people have access to, lots of these kind of conversations where people can go and educate and empower themselves.
Yeah, that would be great, because this is such a deep area, and in short time we just can touch things. But thank you so much, and as always, I learn a lot, and I thank you for your work. I mean, really, it's just so important that people are combining the science, and keep then delivering products that are based on the science. So thank you.
Thank you. It's my pleasure, and thank you everybody for listening.